Matt asked me to speak about genomics, sort of broadly. This is a talk similar to one that I gave maybe at BIF a couple of years ago in Calgary. It was framed towards the commercial producer. Why do genomics matter for a commercial producer? This is also similar to something that we did, sort of a tour around Kentucky and Tennessee cooperatively with the folks at Kentucky Extension, talking to Seedstock producers last year. So I tried to pull those two things together and really focus on why do we care about genomics, if you're utilizing them, what good are we getting out of them, but particularly if you're not, why do we need to integrate genomic testing in a more wide-scale way into the breed? So I'm going to jump right in here and talk really briefly about a couple of really overlooked pieces of genomics, right? So we talk a lot, almost ad nauseum, about genomics number 3, right? How do we genomically enhance our EPDs, make them more accurate, particularly for young animals? I'm going to spend about 95% of this talk talking about number 3. But I don't think that we can overlook the importance of genomic testing on what it has done for wide-scale parentage verification. Having a correct pedigree in a breed is really, really important, right? And these genetic evaluations that IGS runs, if you're a shorthorn breeder, any other breed, right, they still rely very heavily on the pedigree connections that exist throughout a breed, all right? So if those are wrong, we've gone to the effort of collecting phenotypes that we're not leveraging in an evaluation. So parentage verification remains a really important piece of the genomics equation. A genomic test, though, has just made that go from being a sort of standalone thing that we had to do to something that we can capture in the process of generating these genomic enhanced EPDs. And then genetic defect testing, no breed is immune to genetic defects popping up, but genomics has allowed us to, as these defects come online, as we determine what they are, if we're proactive about them, we can get these test markers on a genomic test and make sure that we're able to control for those genetic defects that pop up over the course of time in the seed stock population. So these are the 3 big pieces, but really want to talk just a second about genomics and parentage verification, right? This is nothing more than the process of elimination. We've got a fixed set of parentage markers that we've used for a really, really long time. Again, these used to be standalone, what we called microsatellite markers. They were a pain in the butt to generate. They cost about the same as what a genomic test does now. With the advent of these higher throughput, cheaper, more affordable genomic testing platforms, we're able to put these genomic verification of parentage right alongside all of the markers that we use to enhance a genomic prediction. And then genetic defect testing, again, something that you're all familiar with as seed stock producers. But the thing that I'll sort of, in the context of genomics, finding these things is a little tricky. I work with Dr. John Beaver at the University of Tennessee. John's one of my closest colleagues, and he is the godfather of identifying, mapping, and developing diagnostics for these genomic defects. And John will say that the best thing a breed association can do as these pop up is be proactive. Because right now we're in a situation where we can get these onto the next assay that rolls out that we're doing genomic testing with overnight, right? This isn't a 5-year process. This is if a breed association is proactive about identifying carrier animals. If they're reporting these affected animals, we can find those carriers. We can map these things, I don't want to say trivially, but in pretty short order and have a diagnostic out very, very quickly. So genomics has allowed us to do that. This is no longer a multi-year process to getting tools in the hands of producers to breed around these, right? We don't need to throw the baby out with the bathwater when it comes to genomic defects, and we have the diagnostics, the ability to make them respond very quickly as new things pop up in a breed. The next sort of level of all this is something that some breeds, and particularly the dairy folks, are becoming more and more attuned to, is that there are these things that we call lethal haplotypes, right? These are mutations that you don't get this curly calf that comes out, or this developmental duplication, that extra leg growing out between the shoulder blades of that calf, right? These are just pregnancies that don't happen, right? Fertilization occurs, development starts, but for whatever reason, between that time point and when that calf would be born, something happens to that developing embryo and it disappears, right? The inheritance mode of these is very similar, right? They tend to be recessive, so you have to have 2 copies of the mutant allele for these embryonic lethals to occur. But the issue is that they're a whole lot harder to figure out, okay, this cow lost a pregnancy due to having an embryonic lethal that she was homozygous for versus, she stepped on the wrong anthill, right, and she aborted a calf, right? So teasing these things out is really hard on an individual animal basis. But what genomics has allowed us to do, once we get bigger and bigger data sets, is we can start to look through and say, all right, what do we never see as a homozygote in the population, right? We maybe have 2 homozygous alleles. We see a handful of heterozygous animals. But for this other allele, there are no homozygotes in the whole population, and we've tested enough animals to know that that's not just due to dumb luck, right? Those are where we can start to look for these depletions in homozygosity and start to map and develop diagnostics for these embryonic lethals. So this is some proportion of the decrease maybe in fertility that we experience on the individual animal level is due to these recessive alleles matching up and causing that embryo to abort anywhere from the day 2 all the way up to somewhere in the middle of that animal's pregnancy. These things can pop up. The dairy industry has done a really good job of identifying these. Due to genomics, right, we find the homozygotes that don't exist. We go through, we figure out the biology that might underlie these, and we develop diagnostic tests for them. There's something like, 20 or 30 of these that have been identified, verified in the Holstein population. Diagnostics with genomics can get turned around fast, and now they have a tool to select or make matings that avoid these creating of homozygotes that could interrupt development and cause these embryonic lethals. So again, this is just something that's on our radar in the research community and in some of the larger breed associations, particularly dairy, that has challenges with inbreeding that we're able to start to do with genomics on the population level. All right, so I did my lip service to the 2 purposes of genomics that I talked about first. The rest is going to be helping us dive into what we call these polygenic traits, right? So for the traits that we can't draw a nice, clean Punnett square for, all the traits that are economically important to us that Chip just got done talking about, carcass weight, fertility, anything that touches our bottom line for the most part, aside from coat color and horned polled in these handful of genetic defects, is going to be a complex trait. So we've got hundreds or thousands of genetic variants that are controlling variation in these traits. And we've developed over the course of time EPDs, these selection tools that allow us with some accuracy to predict an animal's genetic merit for performance across these traits. So we're going to focus in on polygenic traits. The challenge here, right, is that a single SNP test, right, we don't have the gene for fertility. We don't have the gene for growth, carcass weight, marbling, tenderness. I just was on the phone call with a producer a minute ago who called and asked about the tenderness markers, right? Turns out, right, that these traits are all complex. Those markers are probably only capturing a small amount of the genetic variation that exists in those traits. So we have to develop machinery that allows us to capture that information and predict these forward when we've got thousands of different places in that animal's DNA that are controlling these traits. So genomics are the tool that have emerged in the last decade and change that allow us to do that with a little bit more accuracy, right? And they're an essential tool for sire selection. I say this everywhere all the time. A tool is only as good as what goes into it, right? If we build a genetic selection tool on faulty data or not enough data, the tool's not going to be nearly as effective, right? Just because we have an EPD, just because we use genomics, if the phenotypes that back that up don't match, it's not going to be a particularly useful tool. And these tools in general, EPDs at the end of the day, they're very, very useful tools. I would argue some of the most useful tools that we have as cattle breeders, but they're not be-all, end-alls, right? We still need functional cattle that perform. There are things that we don't capture with EPDs, right? There are things that a bull has to do, right? A bull has to be able to walk around and mount a cow and breed her, right? There are things that we don't capture in EPDs, so we have to use these as tools alongside all the conventional animal breeding, animal evaluation that we've done for a very long time. So with genomics, right, there's sort of this evolution of the genetic selection tools that we've had around for decades, right? And genomics they do some things and they don't do other things. And I think that sometimes we get a little bit confused, or we make them out to be more than what they are, right? But what genomics do, and this is similar, you could say the same thing about EPDs, right? They help us estimate an animal's genetic merit. These traits are not 100% heritable, right? If you've got a 30% heritable trait, genomics, even in the best genetic evaluation in the world, is only going to tell you 30% about that animal's eventual progeny phenotypes, right? There's still that environmental component that you aren't capturing in a genomic test. This is particularly true with the commercial genomics that are coming online. I think expectations are a little inflated. These are not individual phenotypic predictions, right? What they're doing is they're helping us move the population forward. As a geneticist, I need lots of numbers, right? And I can chalk almost anything up to being on one side or another of that little bell-shaped curve, that normal distribution, right? What we're doing with EPDs, what we're doing with genomics, is we're not doing anything on the individual animal level. Even though we get results back, we test on the animal level. What they're helping us do is move that mean, that average of the population forward. And again, these are not going to capture those non-additive effects, right? So we start to talk about crossbreeding and heterosis. We're not necessarily capturing those in an EPD. Just the heritable additive component of an individual trait. But I'm gonna use this exact same slide in a talk this afternoon on some work that we do, actually looking at how producers, our commercial producers, use EPDs, right? We have worked with some of the folks in our Ag Econ department, and it's really, really surprising when you actually sit down and ask a commercial producer to look at EPDs, and you can follow where their eyes are going, and you can ask them what their decision would be. You can ask them what they would pay based on something that you've set up in an experimental context. It's really, really interesting. So I'd encourage you, if you're interested, I'll talk a lot more about that. But because I hang out with economists so much, I end up framing a lot of this EPD talk, genomics talk, whatever, in the context of risk, right? This bull buying decision is really a large risk that a commercial operation takes on. Investing a large amount of money, right? Bull sales have been really good this year, right? That's still a large amount of money, even when the market is strong, on an animal that's never bred a cow in its life before, typically, right? There's a lot of unknowns that go with this bull purchase. So what we're doing with genomics, what we're doing with EPDs, is we're doing risk mitigation, right? How do we take a little bit of the guesswork out of making this very, very important decision? This bull is going to sire hopefully 100 plus calves on a commercial operation. If a commercial producer gets this right, he gets to reap the benefits for, not just this generation. If he's breeding to a shorthorn, he's probably going to be keeping daughters, and he's going to reap the benefits of a good decision for years to decades in the future, right? If he gets it wrong, it can screw things up in a hurry, right? So this is a risky decision. EPDs, genomics for the commercial producer are risk mitigation. They help us take a little bit of the guesswork out, make us more confident that that upfront investment is actually in what we need to move our operation forward. So I talk about genomics doing two things, right? We're buying these unproven bulls. They prevent train wrecks, right? I grew up on a Charolais operation, so I'm no stranger to a calving season train wreck, right? That bull that was supposed to be a calving ease bull was not, in fact, a calving ease bull, right? And you've got heifers in the barn, and things start to go sideways, right? EPDs help us mitigate that risk, right? That we're going to end up in a really, really bad situation. Maybe it's something like daughter productivity, right? Longevity, stability. You go and make an investment in a bull, you keep his heifers as replacements, you invest all the money in developing them right, you breed them, they get 2 seasons in and things go sideways with their feet and legs, things go sideways with their fertility, trying to get that first breed back done. That's a train wreck, right? Because you've invested not only in that bull, but in his daughter's time and money, and you end up with a train wreck. EPDs and genomics in particular, when you're buying young, unproven bulls, help us mitigate the risk that things are gonna get off the rails there. But what they really do from a seed stock perspective, right? our goal as seed stock producers should be to be making consistent genetic progress, right? Driving traits that we have designated to be important forward. If we're not making genetic progress, what are we doing, right? What genomics allows us to do is to make the right decision in an oriented breeding goal, make that right decision more often than not, right? And hopefully, if our goals are aligned with profitability for our commercial customers, that makes us a more profitable operation as a seed stock breeder, right? We want to identify those little differences across a whole bunch of traits and propagate those genetics forward if we can. So this is the first equation of the day. I'll try and keep it to just this one because it's really, really important, okay? This is the breeder's equation. We build everything that we do in the animal breeding world off of this. What this says is that the amount of genetic change that we make in a population is basically determined by 4 things. And genomics plays a really, really big part in 2 of these that we're going to talk about today, right? So genetic progress is a function of how accurate is our selection. And this is what we'll spend a fair bit of time talking about. So if we have a selection metric, that might be, so say I'm trying to select for weaning weight, right? I can look at a calf and say he looks bigger than that calf. That's not a particularly accurate selection metric. I spend a lot of time in East Africa working in Kenya, and that is how they measure things, right? That calf looks bigger than that calf. We're going to propagate the genetics from a look. If they had a scale, right, that would make that selection metric even more accurate. We're actually measuring that phenotype at a fine scale. If we get a genetic selection tool in the form of an EPD, that accuracy continues to go up. We enhance that with a genomic test. All of a sudden, that accuracy, which is a major driver of how fast we can make genetic progress, continues to go up, right? So accuracy is really the basis of what we're doing as we enhance these EPDs with genomics. They're helping us make the correct decision more often than not. Then intensity of selection and genetic variation get overlooked a fair bit, but on the seed stock side of things, this is where the industry's genetic progress is happening. We can put a lot harder intensity of selection on a herd than a commercial guy can, right? They're buying a bull from us, one bull that's going to sire, 30 plus calves a year, right? That's intense selection on the male side of things. They could never dream of generating that much genetic selection pressure by just selecting the right, 10 replacement females to come into production each year, right? The intensity of selection in the seed stock industry is much, much higher than in the commercial sector. And then we've got to have genetic variation on which to select. If we don't have genetic variation, it's really hard to move things forward. And then the last thing is generation interval. So what's the average age of our parents? In other words, how fast are we able to turn generations, right? If our goal is to make this generation better than the last and the next better than this one, what's the distance between those things, right? That's why we have that triangle G, delta G divided by the year, right? We're living in the real world. If we're slow at making genetic progress, if we're slow at getting that next generation of cow, that next bull into production, that slows down our rate of genetic progress in the real world. And so, again, you guys are all familiar with EPDs, right? They divide out the environmental variation that plays into a trait from the heritable genetic variation and allow us to try and make selection decisions that are just focused on what an animal can pass down to its offspring. One of the challenges I think that I have with commercial producers as I talk about EPDs is there's this desire still, even today, to make selection decisions on a bull's actual performance, right? On a bull's true weaning weight, a bull's true yearling weight. And I know that I'm not the first extension specialist at the University of Tennessee to talk about EPDs, right? But we're still working through this, all right? A bull's actual phenotype, he can't pass down all of the environmental pieces that have gone into making that happen, right? Comparing those phenotypes across different operations is even more problematic, right? It's one thing to do it within a farm, but you go to the University of Tennessee bull test and you start making selection decisions based on an animal's actual performance, knowing that there's all sorts of different things that have happened within his contemporary group that are feeding in to that actual measure of weaning weight, right? Or actual measure of yearling weight when you start to compare those bulls. So I tell our producers, we want to make decisions that are just focused on what an animal can actually pass down in genetic potential to its offspring. So again, the big piece of this that I talk about with commercial folks is how do we get rid of that contemporary group effect? How do we get rid of environment? But the next step, right, and it's what I want to talk about a fair bit today, because genomics is really, really important in this context, is it's not just isolating that genetic component, but it's figuring out which genetics an animal inherited from its parents, right? As seed stock producers, as folks, particularly if you use embryo transfer, right? You generate a bunch of flushmate siblings, you get the EPDs back, particularly once they're genomically enhanced, and they can be wildly different, right? There can be a huge amount of variation in the full siblings, right? From the same flush can have a large variation in their EPDs, and that's not just because Matt's making up random numbers on his random number generator at American Shorthorn, right, Matt? Right, okay, yeah, no, that's real, right? And I want to dig a little bit into the fact that that's real variation in genomics rather than a traditional EPD allows us to identify that early on so that we've got an idea of those flushmate brothers, which ones do we really want to push, and which ones maybe got the bad shakeout of parental genetics, right? So EPDs, right, we're comparing these estimates in an animal's genetic merit compared to another animal, right? These are statistical estimates. They're as good as the data that we have to back them up, right? More progeny records makes that animal's EPD, his estimated genetic merit, compared to his true genetic merit, which we can never know, those bars of where those animals' actual genetic merits might be, they start to shrink down, right? We get more and more confident that the EPD is indicative of what that animal actually inherited from its two parents. Traditionally, that's happened through increasing progeny records, right? We have, a couple hundred progeny on a bull. That EPD accuracy goes up. Our confidence, right, in where that EPD is relative to the animal's true genetic merit goes down. We get a lot more confident. We lower the risk with making a decision between those two bulls because of our confidence in their EPDs. A commercial producer almost never, has the benefit of buying a bull with 200 progeny records in a genetic evaluation, right? So this is where genomics comes in, right? And what genomics allows us to do is leverage this idea of Mendelian sampling, right? That's our scientific term for it. It's just the random shuffling of genes that you get from a parent, right? We've got 30 sets of chromosomes in cattle, and you're independently inheriting one of each of those pairs of chromosomes that your parents has. And what that means is that these siblings, right, even though they each share 50% of their DNA with their sire, they're in red and yellow, 50% of their DNA with their dam in pink and blue, which of those grandparents they're more related to can vary pretty wildly, right? Some of them are exactly 25% related to each of their grandparents, and that's the assumption that we make in a pedigree-based genetic evaluation. You're exactly 25% related to each grandparent. You're exactly 12.5% related to a first cousin, right? But what we see when we start to dig into their actual DNA, this is what genomics allows us to capture, is that there's a fair amount of genetic variation even between these siblings just due to the fact that some of them got more of this paternal grandsire and less of the maternal grandsire. And depending on the genetic merit of those bulls, that can result in a fair bit of genetic differences in genetic merit between those full siblings. This is just an illustration from my PhD advisor, Jared Decker, that he made, basically showing all of the different combinations of chromosomes that can go into a single sperm cell. So over here you've got a blue and a pink set of chromosomes in a bull. The blue from his sire, the pink from his dam. Basically, there's like 1.5 billion different combinations of chromosomes that can end up packaged in one of these sperm cells, right? A huge amount of genetic variation that's just built in to how these sperm cells divide, how chromosomes are partitioned into them, and this is all completely random. So if this bull's sire is an all-world superstar and his dam is a bottom-of-the-barrel animal from the breed, I want sperm cells that are going to be fertilizing eggs that have more of the sire's sire, right? That's the high genetic merit. If you get a sperm with all of his dam's chromosomes in there, probably going to be a lower genetic merit animal. And so if we break this out, I think that this is probably the most important equation of today's, right? That an animal's genetic potential is a function of his sire and dam's genetic potential, right? And then I call this the dumb luck term, right? And that's really all it is. We can generate a huge amount of genetic variation just due to that randomness and the genetics that an animal receives from its sire and dam. And so this Mendelian sampling term, when you sit down and you work the math out, depending on how related the sire and dam are, this can account for over 50% of the genetic variation in a complex trait, just due to that random shuffling of genes that you get from the two parents. And so, again, we can think about a trait like weaning weight, which is maybe 0.3 heritable, right? So 30% of your calf crop's phenotype is going to be due to genetics. 15% of that, so half of that 30% heritability, can come from just the random shuffling of genes in those two parent breeds, right? This is a huge amount of variation. We want to control for it, and that's where genomics sort of comes in and helps us through all this in making these decisions a little bit less risky, because we're able to objectively calculate and account for that Mendelian sampling. So genomics, again, allow us to directly observe the genetics that an animal inherits, right? There are a couple of different ways that we can apply genomic selection, right? So you get a genotype back on an animal, sort of two schools of thought about how you apply those. But in general, what they're doing, and the most important function, is that in an unproven animal, right, a bull that has never bred a cow in his life, we can see a similar increase in accuracy in that bull's EPDs compared to a bull who had a full calf crop that entered back into the genetic evaluation. So a commercial producer has got the benefit of a full calf crop's worth of information, a full calf crop's worth of increased confidence in that selection tool that they're using when they go out and make a bull purchase. So these EPDs are allowing us to reduce risk, but a genomic test allows us to reduce risk, particularly on those young, unproven bulls. You guys are familiar with how all of this works, right? A DNA sample, TSU is probably our most common one now. We observe a number of, we call these polymorphisms. So these are places in the genomes of cattle that we know are consistently different at pretty high frequencies, all right? So what this test looks like is probably somewhere between 50 and 100,000 of these markers. People that are way better at math have figured out the actual math of how you take this molecular genetic information and turn it into relationship metrics between individual animals. But ultimately what they're allowing us to do is represent those relationships between even full siblings better than what we would do if we just made the assumptions in a pedigree. So I'll talk just a little bit. I don't want to get too deep into this, but the reason, so cows have got something like 3.2 billion places in their DNA, right? It's a pretty similar sized genome to what humans have, but it'd be really, really expensive to go through and try and genotype every single one of those markers. One, because most of them are exactly the same. You can stick an Angus genome next to a Brahman genome next to a Shorthorn genome, and they're all going to share well over 99.95% of their DNA is going to look absolutely identical, right? We're a whole lot more similar to each other as cattle than different. You line them up next to a human, and it's still going to be something like 70% exactly the same, right? Because we're all mammals, right? So we don't need to look at every single place, and we don't even need to look at every single place that animals are different. Because we take advantage of this idea that DNA gets inherited in chunks, right? You get either full chromosomes or this recombination event sometimes will happen where a paternal and maternal chromosomes swap a little bit over. We'll skip over that diagram. But the crucial part, right, is that we can stick a marker, even if it doesn't affect a trait directly, if we know that it's evenly spaced from other markers, DNA gets inherited in chunks. What we're effectively doing is saying, all right, for this marker, we know that those flanking DNA places in the DNA, there might be a whole bunch of other markers that affect the trait of interest, but all we need to do is estimate how much of a trait's variation is controlled by that marker and allow it to represent a large chunk of the genome, right? So eventually, these marker associations will break down, but that's going to take hundreds and hundreds of years for the markers that we've chosen to lose any association with the surrounding regions in the genome. So genotyping arrays, these have been the workhorse of how we do this. The interesting thing I always point out is that these Australian guys wrote this paper in 2001 talking about how do we do genomic selection, right? And it was another almost 9 years before we actually had the tools to do what they said back in 2001. So really the machinery that we're using was proposed back in 2001. The tool doesn't come out until about 2008 or 9. But what it's allowed us to do is measure, objectively observe the variation in those chunks across the genome and integrate that into the calculation of our EPDs across agricultural species. This is the long and short of things. I didn't pull this in from Shorthorn, but this is from American Angus. These are the progeny equivalents of a genomic test, right? And I think that this is the best way to measure how effective these things are at increasing our accuracy and confidence in an EPD, right? So we've got traits in the first column. There's sort of two versions of this. And then progeny equivalents. How many phenotyped calves out of this bowl would you need to enter into an evaluation to see the same increase in accuracy that you get from a genomic test, all right? So for, again, calving ease direct, that's a whole calf crop's worth of information, 28 progeny equivalents there. Same goes for weaning weight. And then we start to think about some of these other traits, so things like heifer pregnancy, mature weight. These are things that are more expensive in both sort of time and inputs. These are more expensive traits to measure, but we get a boost in accuracy somewhere between 15 and 20 individuals in those traits. Very, very valuable for a commercial producer as they try and reduce that risk associated with making a bull purchase. I very briefly want to talk about the importance of phenotyping. All right, I think that in a lot of other segments, genomics got sold as a be-all, end-all, right? We have genomics. We don't need to phenotype anymore, right? We have the tools at our fingertips, right? That is absolutely not true. If we stopped collecting phenotypes as a breed association, the connection between animal phenotype and genotype and pedigree would erode over the course of a couple years, right? There would be an immediate decrease in the usefulness of those genetic selection tools if we stopped collecting phenotypes. And I would argue that in a lot of breed associations, we maybe have enough genotypes to be useful. Angus they legitimately throw out about 80% of their genomic data in the method that they use because it's just computationally impossible to put to use, right? But they would sure like a whole lot more phenotypes, right? It doesn't make as big of a difference on an individual animal, right? But on the genetic evaluation scale, we're in a pretty good place with genomics, but we've got to have a connection between them and phenotypes. We have to consistently keep genotyping animals, but more importantly, we have to keep phenotyping animals. So the two sort of models that we utilize across the industry, single-step GBLUP would be sort of the Angus model. What that's doing is effectively making a genomic relationship number that's different from that pedigree relationship number that we assume between animals. The way that things work at IGS, which is what I'll talk just a little bit about at a surface level, is this hybrid marker effects model. So this is a model that's actually going through with a subset of markers that we've identified as associated with traits and estimating the effect of having a genotype at those particular markers. And then coming back through on the back end and integrating with pedigree, right? So the pedigree that you've put together at Shorthorn, it allows us to integrate those in and correct and pull in information from animals that haven't received a genotype to help boost their accuracies as well. So the gist of this, right, and again, I won't go through it in any great detail, but we go through each one of these markers that show up on our genomic test. We estimate the effect of having an A or a B allele, right? So we've got two versions of a gene at this position. We estimate the effect on a phenotype of having that version. We do that across all of the different markers in the genome, and we do some simple addition. and we end up with what we call sort of a molecular breeding value, right? So what's the effect of having all of the markers in this animal's genome, and then come back through and do some pedigree corrections, pull in extra information from across the pedigree, and what we settle on is a difference between these animals sort of in genomic plus pedigree space. Again, math that guys way smarter than me have developed, but the long and short of this is that no matter what genetic evaluation you've got cattle in, from a theoretical standpoint, they should all converge on about exactly the same numbers, right? This is not terribly different from a conceptual standpoint from what Angus is doing if you make a couple of assumptions that we're pretty comfortable making in the genetics world. All right, so this is the one thing that I'll leave you with in regards to GE-EPDs, and then I'll maybe walk through one example, but it's so important to remember that regardless of whether a genomically enhanced EPD improves the EPD's value or makes it go down, right? It's always better than an unenhanced EPD. And I bet Matt could attest that you never ever get calls when EPDs improve when a genomic test comes in, right? I've never got one either, but I've gotten a whole bunch when they've gone down, right? The long and short of it is that the genomically enhanced EPD's increase in accuracy gets us closer to the true genetic merit of that animal, right? And being closer to the true genetic merit of an animal is always better, right? It allows us as seed stock producers to make better decisions. It allows our customer to make a more informed decision. He's buying a number that's more representative of what that animal is actually going to do in his herd, right? So that increase in accuracy gets us closer to truth, and that's always better, right? Understand that some are going to go up, some are going to go down, right? We make assumptions when we have a pedigree estimated EPD about how related that animal is to everybody else, right? The genomic reality I just showed you can vary a ton. We can go from 3 flush mates looking exactly the same with a pedigree-based EPD to 3 flush mates looking wildly different once we add genomics. But I can be very confident in saying that that flush mate that comes out on top after the genomic test is probably the one that we want to propagate into the population in subsequent generations. So just a quick example here to show when a breed leans into genomic testing, basically overnight, the difference that it can make on 2 big pieces of that breeder's equation that I showed earlier, right? So I talked about accuracy of selection. I talked about generation interval. So the dairy industry is a perfect example of when you lean fully into genomic testing, what it can do to both of those and how that helped them drive genetic progress really through the roof when genomics came online, right? So a couple of things here to think about as we consider accuracy. One, how much is accuracy worth, right? That's a tricky one to put your finger on. For me, it's hard to place an economic value on accuracy. A lot of that is just risk tolerance, right? How confident are you in being right or wrong about this animal, right? How confident that bull with a 0.2 accuracy on calving ease direct, how confident am I that I want to put him on heifers, right? So again, accuracy is a tough one. And then where is that accuracy most useful as you're going out and making a sire selection decision is important to ask. But particularly, what would you sacrifice in terms of genetic potential to get accuracy? All right, this is a question on some of the behavioral work that we do with our economist friends. We always ask people their risk tolerance, right? And this is really a risk tolerance question, right? Are you comfortable sacrificing genetic potential? Are you comfortable just using that proven bull that's maybe 2-3 years behind genetically, all for the sake of being confident in his EPDs, right? So these are things we have to keep in mind. Genomics, though, in my mind, allows us to hedge some of these, right? We can get the best of both worlds. We can not sacrifice genetic potential. We can breed that year's hottest bull, but with the added benefit of being a little bit more confident that his EPDs are representative of his true genetics. So this is where the dairy folks sort of ran into issues for a long time, right? I can use a young bull who's got low accuracy, but we've had another generation maybe of selection we've turned over. This is the potential for having the hottest genetics in the entire population, right? Quick generational turnover. We can also just use proven bulls, right? So we only want high accuracy bulls. We're confident that their EPDs are representative of the genetics that they're going to pass into our herd. But the issue, right, is this bull's maybe 3, 4, 5 years old. And we're just now getting confident enough in his EPDs, right? Again, dairy sire testing is how we got around this pre-genomics, right? So this is sort of the sequence of if you were a dairy bull calf who was maybe going to enter into an AI stud, this is the path that you take and how long it goes, right? Bull calf born on day 0, right? So we wait for the bull about a year, right? We can finally collect semen on this bull. So he's a year. We breed that bull to a group of test females, right? And then we stick that bull in a pen somewhere and make him wait, right? So we have to wait another 9 months for those heifers that we bred with this bull to get born. Half of those calves are female, right? So we throw half of them away. And half of them actually have the phenotype that we care about measuring in the dairy industry. And so we wait for those calves to grow up, right? We breed them as yearlings. And then we wait for them to gestate for 9 months, and then they enter their first lactation, right? So we've just waited 4 years with this bull parked in a barn somewhere to figure out if his pedigree estimated EPDs are of how indicative they are of his actual genetic merit, right? We get this daughter's first lactation information, flows back into the genetic evaluation. We see an increase in accuracy. 2 of those 50 bulls go to an AI stud. The other 48 go to McDonald's after we fed them for 4 years, right? And this is bad for generation interval, right? Because all the while we're making genetic progress in the background, but we're just waiting on these bulls for 4 years to figure out if they're actually going to enter into be elite sires in the industry. And so genomics overnight transforms the dairy industry because this generational turnover is a huge problem, right? We have a bull born now. They get a genomic test somewhere in the first couple months of their life, probably in that first week. The elite calves go to a stud. The non-elite calves go to a calf farm somewhere and then on to the feedlot. And we've got daughters in production of these bulls within a couple years, right? The beauty of this, they outline really nicely in this paper. I give this to all my grad students to read because it's a perfect testament to what genomic selection, when you implement it right, can do. And so what you're looking at here are 4 different subpopulations of dairy animals. The blue and the red are the sires, and that's their generation interval, right? So the average age of a sire, an American Holstein, in 1990 is about 7.5 years old, right? We have basically no change, right, all the way up to 2008. The average age of a Holstein sire is still about 7 years because they're having to go through the progeny test. Those bulls, before they actually become productive AI sires, have lived the majority of their life not contributing to the population. But what happens in 2008 is genomic selection comes on board, and immediately that sire testing goes away. And now, and this trend has continued down a little bit, there's obviously a limit, but the average age of a Holstein sire is 2 years, right? We lopped 5 years off of the generation interval of our bulls in the dairy industry just through the utilization of genomics. And that is really, really powerful, not just on driving generation interval, so we're making genetic progress faster, but across all of these traits, you look at the inflection point that happens at about 2008, that accuracy of selection and the generation interval combined takes the genetic trend lines for these and puts them on a completely different scale, all right? So genomics are a really, really powerful tool when we as a whole industry buy in and utilize them in a productive manner. All right, I think that's all I've got. Maybe you have time for a couple of questions. Some takeaways there, my contact information, and I'll be around for the rest of the afternoon. I appreciate Matt for bringing me onto the program. You've got a really, really good thing going on here, so happy to be north of the border in Kentucky talking about genomics. So thanks, Monty. Thanks, Matt.
Thank you, Troy.